Before we start with today's seminar, a small advertisement for tomorrow. In the name of all members of IFISC working in photonics, we would like to invite you to attend a workshop that we are organizing tomorrow. And the workshop is entitled New Trends in Photonics. And we have been able to win two very high-level speakers from the US for these workshops. One is Ken Choquette, who's from the University of Illinois, and the other Kun Sheng from the Arizona State University. Both can be considered real pioneers in the field of photonics. And it's a unique chance to interact with them tomorrow and to attend the talks. So we warmly recommend you attending. The seminars on the workshop will start in the talks here in the seminar around 9:30. If you're here a bit earlier, that would be nice, and we will finish just before lunch break. So it will be here in the seminar. So hope to see you.
Thank you. Before we start also the seminar today, Happy New Year. As always, introducing new people at the Institute, Lucas Lagasa, that arrived last Monday. So we working here as a also physics. Second short notice, after the end of the seminar of today, we will have coffee here inside the room. We will show multimedia presentation, which was the Happy New Year message of the President of the city, which is good, which was very good.
Well, I think that probably you know already Ernesto, but as he has been here only for one and a half months, I mean, still Christmas and so on. Let me say that Ernesto Nicola has a strong links with Dresden from where he's coming now. He started for his PhD there with Marcus Baer, basically on pattern formation, spirals, and all these kind of things from the point of view, strongly from dynamical systems, contribute to the continuation techniques and so on. Then in a number of postdocs that have been for 2 years with our friend of many of us, Jaume Casadepunti in Barcelona, he has studied and continued studied some problems in pattern formation, related also to chemistry, right, chemical reactions. Then later was for a year in Germany, and finally for a few more years in Dresden. And then his research topics have changed strongly to the kind of lecture he will give today. So in biophysics, with both strong connections to experiments on the one hand, and he will explain, but also to theory. So I think it's.
Thank you, Manuel. Welcome everybody. Actually, I'm here since 2 months, and before, as Manuel mentioned, I was almost 3 years working in Dresden, in an institute where people mainly do biophysics. So and there I shift from nonlinear dynamics to actually nonlinear dynamics, but applied to biophysics and cell biology. And there I started and I continue working in a number of different projects, and I just wanted to give you a very brief overview. in a couple of minutes, just so that you get a feeling of which kind of things I do and what I like and what I really could do. And after that, I will go into the topic of this talk. So essentially, this stuff that I've been doing in the last couple of years can be classified in three, depending on the tools that I use to analyze, in three different groups. And the first group, which is pattern formation, which is something which I've been doing since a long time, and since more recently I've been applying that to biophysical problems. There I've been working with the modeling of spiral dynamics in glycolysis, which is a chemical reaction which occurs in every cell and is very important for the metabolism. And the project that I will tell you today, so I won't go into more details on that. Both projects actually are in collaboration with experimentalists. Then I have an array of different projects in which I applied more, not pattern formation, but more basic bifurcation theory or really nonlinear dynamics, and also more lately bifurcations in delay differential equations. And those projects include things like signaling networks or effects of time delay in gene regulatory networks, effects of inertia on hair bundles, which are some cells which are responsible for hearing, and also something related to muscle, flight muscle of insects. And before, actually, I was working on modeling of the regeneration of Hydra, which was a very nice project, but essentially I'm not working that anymore. And also I've been doing a couple of things which are more related to stochastic processes. One of them is related to the cooperative motion of molecular motors, and another one which is more recent which is about simple models of swarming behavior. And so now let me jump into the topic of the talk. And before I begin, let me mention, well, I already said that this was a clear joint experimental theoretical collaboration, and the experiments have been really remarkable here, because all the modeling that we had, I mean, the complete problem arises from the biologist side. So they proposed us if we could model something that they have, and they knew many facts, and you will see how the story goes. And so the experiments were done in Dresden, Germany, in Max Planck Institute for Molecular Cell Biology. Were done mainly by Nathan Goering, which is this guy here, which is a senior postdoc, and he's working in the group of Anthony Hyman, which is one of the directors of this Max Planck Institute. And from the theory side, I teamed with Stefan Grill, which is both at this Max Planck Institute for Biology, but also in the Max Planck Institute where I was, which was for physics of complex systems. He has a joint experimental theoretical group, and with him we have been doing the modeling, and with him and mainly with a master student, Philippe Couton, which has been working with us for 1 year. So let me first briefly outline the structure of the talk. So I will give you a rather long motivation of the problem. Then I will introduce the model that we have been studying, or that we presented, and then I'll go into the analysis of the model. Finally, before the end, I will come back to the connection to the experiments, and I will finish the talk with a short summary. So, the motivation. So, cell polarization was in the title of the talk. So, what is cell polarization? Essentially, cell polarization is nothing else than what us physicists know as symmetry breaking, but in the area of symmetry breaking in single cells. So actually the idea of symmetry breaking probably is familiar to most of you in systems, in physical systems or chemical systems, but also occurs in many situations in biological systems. And that can be either be at the very small scale of one single cell, which has a broken symmetry, or it can be at the level of one organism or more or societies. And in the level of single cells, biologists call this symmetry breaking polarization then. And so probably you realize that most cells are actually not symmetric. So most cells, you can say that are in one way or the other polarized. And it's a very hot topic in biology now. The mechanism by whom a cell breaks the symmetry, and there are actually a number of mechanisms that are behind the symmetry breaking. And in particular, in the case of polarization, there is a number of cases which biologists have studied, here I just list some few of them. For instance, in the case of asymmetric cell division, I won't go in detail into this now because this is essentially the topic of this talk, so I'll come to this later. Also, symmetry breaking occurs in the case of migrant living cells. Also, epithelial cells, so the cells of your skin, are clearly asymmetric, and there is also a polarization process which occurs during the generation of the skin cells. And also the neurons are, of course, asymmetric, and the way that they get asymmetric is also through a polarization process. And we will see in a few slides that all these cases here actually share some mechanisms, and so that's why it's so important to look very carefully to some of these cases, because for us physicists it's probably not so obvious, but there are some commonalities in biology. So there are some mechanisms which are really occur all over, all the time. So, and now biologists are getting so deep in the knowledge of the molecular cell biology that we are now able to discover which are those generic mechanisms. So, before going to the polarization in asymmetric cell division, let me just show one particular case of polarization. For instance, in the case of neutrophils, which are some cells which essentially are more or less symmetric, and then when they feel a gradient, so some chemical gradient by the mechanism of chemotaxis, they are able to rearrange themselves and travel in the direction of the chemical that they are sensing. And this mechanism by whom they decide in which direction they want to go is actually a mechanism of polarization, and there are some proteins which are responsible for that. And
but this is thermophos, which is the chemical gradient.
Yes.
So there's no
but even if you put, they can also spontaneously break the symmetry from time to time. There is a one has to distinguish two different cases. The case where you put some little signal, and then the system is able to sense that and polarize or get asymmetric. So it's in a way a kind of amplification of the little signal that you put. And you have other cases where the symmetry breaking occurs actually spontaneously.
But this is not the case.
This is not the case.
Go back.
Yeah.
So in these figures, in the first is neutrophils, and what's the symmetry that is broken?
It's difficult to see, but the cell essentially is more or less round here, and essentially what happens is the following. It's a kind of cascade mechanism. First you have one protein which essentially gets asymmetrically distributed, and that protein, that's something which is occurring here.
At the gross level, so what you mean is that it is no longer spherical?
Yeah, it's actually moving. It has a so-called pseudopodia. It's like it grows.
And the second figure?
The second figure is Pictosphaerium discoideum, which is a very famous system, and here they externally input a very a weak gradient, chemical gradient, and then they are able to measure the amount of the protein which is able to generate this symmetry breaking, and it's what you see in white here. And then with this star is where the orientation that they put the gradient, and then it's a kind of things that biologists.
That's not spontaneous.
No, did I ever say that these two cases they are not spontaneous? These two.
Yeah, okay, with the seminar we read symmetry breaking, and we assumed it was spontaneous.
I mean, there is nothing very peculiar about breaking a symmetry posing a field, right?
No. Yes. So another case of where sorry.
No, discussion.
Another case where polarization occurs is in the case of the so-called asymmetric cell division. So typically, when the cells divide, either the two daughter cells are equivalent or they may be different. This later case, the one here, is called asymmetric cell division, and it's a very important mechanism for multicellular animals, for instance, for animals, multicellular organisms, because it's one of the mechanisms that allows you to have many hundred different cell types in your body. And so essentially it allows for two daughter cells to have different fates, to follow different lines. And the main organism where this kind of asymmetric cell division has been studied thoroughly is in the case of a very simple animal, a worm called C. elegans, which I will introduce now. So this is also the worm that the biologists with whom I work are doing the experiments, and essentially this is a very small animal. It's about 1 millimeter long, and it looks like a tube and has a mouth here and an anus here, and it essentially has a very thin intestine here, and the rest of the body, like 80% of the body, is essentially dedicated to reproducing. And you see here, these blobs here are actually daughter cells, or cells, embryos, which will essentially later on go out of the mother. And these, and essentially in these daughter cells here, which are here, is where this well-studied case of asymmetric cell division occurs. Indeed, so the life of a new animal, of a new worm, goes like this. So first you have the fertilized embryo, which is just one cell. Then it divides into 2 cells here, then into 4, 8, 16, and so on, until you have the little animal here, which is what goes out of the mother. And what is interesting is that this first division from here to there, so here you see that you have a big cell here and a small cell there. So that first division there, which goes from the situation where there was only 1 cell and goes into 2, is asymmetric. And this is the main system, the main case that where the biologists studied all the details of this polarization process.
Can I make a silly question? Yeah. If I had to split something, probably it's going to be the 2 pieces are going to be different. That's the typical outcome I expect.
Can you repeat?
Yeah, when I split something, a cake or something, whatever, so the 2 pieces typically are going to be different. But so it's very hard to have the 2 pieces exactly the same.
No, I don't mean, so I wasn't clear. These are not only different because of the size. Also, the proteins which are in each daughter cell are completely different. Actually, this cell here will eventually become those so-called gonads, the cells that produce sperm and eggs, and where these other cells will become muscles. So the proteins, you will see that in a moment, the proteins which are present in one cell and in the other are different. It's not just a matter of the mass of the two, that the 2 cells are different. It's just the fate. Mm-hmm.
Yeah, well, but at this level, well, what I understood was that it's just the genetic material that is splitting, right? So you
No.
get
The genetic material is the same.
It's the same.
Yeah.
So you go from 1 to 2, but also you are saying that there is more inside the cell that is also splitting, because I think what you're talking about is cell differentiation, right? What part?
Yes and
the
no.
cell is going
The
to
cell
become.
differentiation is a very general term. You can have so, for instance, one way in which you can get, say, a muscle cell and a neuron is that first it divides symmetrically, so you have two equal cells, and then these cells sense an external chemical which makes them take a decision, take a different path. But when they were born, and when they are born, if you reverse the chemical gradient, the fate will be also reversed. This is different. This is already, the symmetry breaking in a way happens before the separation between the two cells has occurred. Is that clear?
Yeah, more or less. Well, my question was, I think the question is more why the cells split the way they do, more than, because, let me see if I say the things in there. For me, it's more complicated to do two pieces exactly the same, with the same amount of whatever, than to do it in a different,
Actually,
in an asymmetric way, but it's not that you do it in an asymmetric way, but it has to be exact, always, if you pick another C elegans, it has to split exactly with the same balance, and this is what is happening. Right?
Yeah.
This is what you have to
But the
understand.
interesting thing now is that this balance, if you want, is not regulated by the genes itself, but this, as you will see, is regulated by something on top of the genes. Probably it becomes clear after later on. So here you have a movie. In this movie, I'm showing. Well, it's just the course of this first cellular division. But now, so let's take a look of what is actually. This is again the one cell stadium, and then divides into 2, 4, 8, 16, and you see here that essentially here with the color is marked which kind of proteins are expressed there. And so essentially this asymmetric cell division determines what these cells here, or this side here, will become the head of the animal, and this other, the right side, will become the tail of the animal. So, but now, so the biologists not only look to the size, but they also they want to see what is difference inside each of these two daughter cells, or even before they separate, what makes this cellular division give birth to two different cells. And what they see is that there are some proteins which have broken symmetry, and in particular they found that there is a family of proteins, the so-called par family, par 1, par 2, par 3, until par 6, which are the proteins which typically become asymmetric, and they start actually from a symmetric situation, and then before the cell division occurs, they become asymmetric, and that's actually the process that we want to model here. And so essentially, what they see is that you have this is just before the cell division occurs in this plane here. They see that in the membrane you have a large concentration of what here is indicated as red, which is a protein called par 6, and in the tail side you have a high concentration of another protein, and almost no of par 6, which is called par 2 and is indicated in green here. And but this is just before the cell divides. And so, but maybe I should say that here I can mention that actually these par proteins which are seen in this animal, in this worm here, are actually seen to exist in many other kinds of cells which can polarize, as I mentioned before, for instance epithelial cells or crawling cells or neurons. Here maybe not all of the members of the part family are present in all these cells, but at least some members are present. So there is some common phenomenology behind the polarization in many different cases. So it's really worth to look more carefully what is going on there. So this here I have a list of the main facts that the biologists know about these proteins, which are responsible for the cell polarization. So what they have seen is that they are able to somehow inhibit the production of many of these proteins, and then by that they can see when they disrupt polarization, then they say, aha, this gene was important. So, and with that mechanism, they have been able to single out two genes which are really important, and which are the ones that are indicated in red here and in green here, the part 2 and part 6. They also know that when they are in the membrane, they tend to repress each other. I will go into more details about this later, but essentially it's like they don't like each other, or they have some kind of competition going on between these two proteins. And they also know that the total amount of protein present in the cell, the total amount of part 2 and the total amount of part 6 is constant, at least during the first stage of these first cellular divisions, because there is no transcription going on during that time. So there is no production of protein. So all the protein which is there is the protein that was there, that the mother gave to these cells. And another important feature that they have seen is that they seem to be important to distinguish. So these proteins can either be in the bulk, in the cytosol, or they can be in the membrane, also called cortex. And they do different things, either if they are in bulk or if they are in the membrane. And these proteins actually can go from the bulk into the membrane and back. So, and another important thing is that this process, how you get to here, is actually dynamics. And the process begins by essentially a situation where you have the part 6, the red protein, on the membrane, whereas the part 2 is essentially all on the bulk. And then, as time goes on, you have that a bit of green going up here, and the red essentially retracting, and essentially until this reaches this situation, and here is where the cell divides, and of course, then the head cells, so to say, gets all the red protein, and whereas the tail cell gets all the green protein. And here I have
you just plotted the protein and the membrane, right? Because the total amount is constant.
So maybe here is seen better. So essentially, as I said, the total amount of protein is constant. The total amount of green is constant. The total amount of red is constant. You don't get distraction of that. But you can see here that you have a red more or less all over the cortex, and green is all concentrated in the bulk. Yeah? And then you see that here you have a higher concentration of red, so means there is some red which is going from the bulk into the membrane. Yeah? And actually you have some red which is disappearing from this side. And you have some green actually that is going there. Yeah? And so this is the process that they see. Yeah? And this situation is what we would like to model. And for that, we will use the facts that I already mentioned. So to summarize the ingredients that we are putting in model are that we will deal only with two proteins, two kinds of proteins. There's called part 2 and part 6, what was green and red before. We will assume a reaction diffusion mechanism for them. I will go in more detail in the next slide about that. As I said many times already, mass conservation, so total amount conservation we assume. And the two compartments which I already mentioned. And the cycling from the proteins between the membrane compartment and the cytoplasmic compartment. And so, more specifically, so we will be dealing. So we have these two compartments, and then say we take the protein part 6. For that, we have a concentration of part 6. I call it B6 in the cytoplasm here, and a concentration on the membrane. I call it A6 here. Yeah? And then, I can write reaction diffusion equations for those concentrations. Essentially, for instance, the concentration of the protein in the membrane is increased as protein from the bulk goes into the membrane. That's indicated by this term. It decreases when the protein in the membrane goes to the bulk. Yeah? That's indicated by this. And since you want to have total concentration of the sum of B6 and A6, you have to assume that these rates K off and K on are essentially the reverse for the description of what is going on the bulk. Yeah? So till here this is trivial, it's just essentially the same equation. But now the key point comes here, that the diffusion on the membrane of these proteins and the diffusion on the bulk is hugely different. It's very different. Indeed, the diffusion on the bulk is like 1,000 times faster than on the membrane. This is rather easy to understand why so. I mean, essentially the bulk, you can consider that it's a rather big protein diffusing in water, whereas that big protein in the membrane is like if it were diffusing, say, in butter, so something which is very viscous. So that is what is behind these high different diffusivities. And essentially for the other protein, for the part 2, you have essentially another 2 equations for the concentrations. So you have overall 4 PDEs that describe the evolution of the concentrations of these 2 proteins. Yeah? And now the interesting point is that the biologists are able, by doing some experiments, some so-called FRAP technique, they're able to measure actually this absorption and dissolution rates. So they're able to put numbers into this K on and K off for both proteins. And they are also able, as I said before, to measure the diffusion constants of these proteins in the bulk, in the cytoplasm, and in the cortex. And as I said before, you have this situation that the diffusion in the cytoplasm is much higher than in the cortex. So this last thing actually allows you to get rid of 2 equations, because essentially, if you go back here, so things diffuse so fast in this compartment that essentially the concentration of both proteins here is space independent in this compartment, but not in that one.
Can you go back a moment to the equations? So the reaction part is linear.
Yeah, but this is not the whole story. The interaction is missing. That comes. That part is what introduced nonlinearity is the interaction between both proteins. This is just like if you have a surface and you have a gas phase and you have some proteins. Yeah? But as I said, the diffusion in the cytoplasm is so fast that you can actually eliminate it, and you can essentially get rid of 2 equations, and essentially you get to this stage. So now you have equations which describe the concentrations of part 6, or the red protein, in the cortex, and the green protein in the cortex, called A6 and A2. And okay, but now what is missing, as Max pointed, is interaction. I mean, otherwise it's trivial, the thing. So now, as I mentioned, the interaction seems to be some kind of they seem to repel each other, roughly speaking. What is actually happening is that, say, when the concentrations are, they seem to interact only when they are in the membrane, Yeah, and if they are both in the membrane in the same place, the one that has higher concentration somehow wins and pushes out of the membrane the other protein. Yeah. So essentially, what it does, a high concentration of one protein essentially enhances the detachment rate of the other protein from the membrane. And essentially, this, and since this mechanism actually, it is so, but it has some saturating properties. So for that, we assume a kind of Hill type function. So essentially, the off rate, the dependence of the off rate for the A2 is very low if the concentration of A6 is low, and increases as the concentration of A6 goes up until it saturates. Yeah, and essentially with this we arise to actually the model that we have analyzed. So essentially, we have two variables, two concentrations for the proteins in the membrane. We have, this is the only nonlinearity, is this a reaction diffusion system? You have the diffusion in the membrane, which was slow, as I said before, but also has here these average terms, which are essentially integral terms, so global coupling terms, right? And and as I said before, we essentially know almost all the parameters which appear. We could measure this from the experiments. We can measure all these parameters which appear in these equations. So now let's see.
The conservation law, is it trivially verified?
The conservation law, somehow you use it to get rid of the second of the two equations. The point is that, yeah.
Still here, A2 and A6 must be constant. The global amount, the integral of A2 and A6 must be constant.
No, let's see. I mean, biologically, what is conserved is the total amount of A6.
This is just the one on the membrane. This is just the one on the membrane.
Yes. But if you can include, I mean, essentially this C2, which is the total concentration of protein minus the average on the membrane, essentially gives you the other one. Okay, so now let's see what kind of things we get from this model. So first thing to do is to look for the fixed points, to the homogeneous dynamics before going before looking to any pattern. So we want to see what we get. And actually, we could have guessed from the beginning what we would get, because essentially, as I said, these two proteins in the membrane, they tend to repel each other. And as is well known, if you have symmetric inhibition like this, you typically get bistability out of that. So, and that's shown in this figure where I vary the total concentrations of part 2 protein and part 6 protein. And as you see, there is a broad area here, this dark gray area where you have bistability. So you can have, this bistability is actually finished, or it ends in a typical cast bifurcation, which is something which we could have also guessed, or which is something which should be generic, because whenever typically bistability is in a two-parameter space, disappears in this way. So now what happens if we add the diffusion and the integral term? Well, the integral term we already had in the homogeneous dynamics. So can we get the formation of domains? And
that solution corresponds to what? That everywhere you have the same density of 6 and 2?
That is homogeneous.
The point of what you are looking, so complete mixing.
Yeah.
Well mixing, so
it's a well mixed case. Yeah. It's essentially when you turn the diffusion on the membrane, you turn it very high, so you put a well mixing there, and you get that. But of course, the system has a rather small diffusion constant, so that will probably favor the formation of domains.
There are another two solutions that, I mean, you have all two in the cytoplasm and 6 in the membrane. That should be also a solution, right?
Look, these are so the two typical cases. So here you have that the A2 is very low in the membrane, so it means it's all in the cytoplasm, and the A6 is essentially all in the membrane. And the reverse is also possible. And so in this area you see this, in that area you see that, and in this area you see essentially both. Yeah. So you have stability between these two possibilities. Okay, so we want to see if the system is able to form domains, under which conditions, and if there are some instabilities behind these domains and so on. So here we have a typical outcome of a numerical simulation. So here we have space and time, and essentially you start from one of these fixed points, but with a little perturbation, and then essentially you have that a domain arises after a rather short time. And here there was a movie of how the domain arises. And now if you go back to this parameter space that I was showing you before, and you plot or you look the areas where you see the domains, you see that actually the domains you see in all these big, huge gray region here, the light gray region, whereas the dark gray was the original bistability. So you see that actually the system, when you include diffusion, it actually is much easier for the system to form patterns, or to, yeah, that actually, or if you want, the possibility to form patterns is much bigger than the bistability region. So, now we would like to understand why, if there is some, what is the mechanism behind the formation of these domains, right? But maybe for doing that in the original system, I will now. So essentially, you can, if you look to the instabilities, you see that there is inside this blue region here, there is an instability going on. So whenever I put my homogeneous state in here, it essentially it builds up a pattern. Whereas outside this blue region and in all this gray region, I have coexistence between homogeneous solutions and pattern solutions, or domain solutions. And so this blue line here is clearly an instability line. I would like to know which kind of instability it is. And as I said before doing that, or rather than doing that in the full system where you don't learn very much, it's probably more, it's better to do it in a simplified system. So we go now from a two-variable model to a kind of normal form or amplitude equation, which is much simpler, but contains all the important features about the two-variable model, the general model. And essentially, to write that, what I do is I essentially go back to the point that, so we know that there was a cast bifurcation, this kind of wedge-shaped area will finish in a very particular point, actually this point here. So, and we know that near this area, there should be a normal form for that, which is essentially given by, this is a canonical form of that, which is the normal form of the cast bifurcation. And to that you have to add then foldings of the diffusion and of the global coupling. So this is, so to say, this is our toy model, and you will see that here in this model is actually much easier to understand things. So, but before doing the analytics, let's explore a bit numerically what this model gives you. So this model essentially has two important parameters, which are this mu0 and mu1, which is mu0 and mu1 here, and essentially you have also a wedge shape area here, the dark gray area where you have bistability, And as you see in the light gray area, you have the formation of domains as well. And also you see here a blue line, which inside this blue line you have the homogeneous state is unstable, whereas outside is stable, but then you have coexistence between domains and the stable state. And you, of course, already see here the correspondence between this is the toy model and this is our model of polarization. And you see that topologically they are, of course, equivalent. But now let's look more carefully what is actually going on, which kind of instability is giving rise.
For you, you wrote before that the A is the difference between the A2 and A6. Is this just
proportional
mathematical, or
yeah.
you can show this from the analysis, or is just something related to it?
You can. I did the derivation. I know the mapping between these guys and the original guys. You can do that.
So it means that there is a mass variable which is the sum of the.
Yeah, it's actually proportional. Yeah.
The total amount is something like a mass variable. So the total amount of A2 and A6 is something like a mass variable, close to the mass quantity.
I can show you how the phase space looks like. It's not so easy. I mean, you have a fast manifold and a slow manifold.
You have
and the
to
fast
them and the slow one is related to this.
Yeah. This is the projection on the slow manifold.
Okay.
But you can do it by there are different methods. You can do it with multiple scale analysis or yeah.
And parameter mu zero just tells you that the
this is essentially a pitch fault.
well, physically it means that the concentration of A2 and A6 are not the same. The global one, I mean, you're breaking that asymmetry. This equation is not symmetric A going to minus A.
You mean here I have a symmetry?
Well, parameter
No.
mu zero breaks symmetry
Yes.
A going to minus A.
Yeah.
And what does this mean? That the concentrations of A2 and A6 are different. The global amounts of A2 and A6 are different.
Mu
Yes.
zero one side.
Essentially, this line mu zero means is essentially this line, and if you map it into the original state, it's a line that goes like this. It's almost a diagonal, but it's not really the diagonal. So it means essentially one concentration is almost the same than the other.
For mu zero equals zero.
For mu zero, yeah, for that particular case.
So the fact that mu zero is different from zero tells that you are studying situations in which the global amount of A2 and A6 are not the same.
Yes. Yes. Yeah. As I said, U0 is essentially the diagonal in this plane, and this is total concentration. So the diagonal is one is equal to the other. Yeah. You're right. Okay. So what kind of instability it is? And in the framework of this equation is rather easy. So you do the typical perturbation ansatz. So you have a fixed point, and then you put this P tilde is a perturbation, which has a wave number k and a growth rate sigma, which depends on k. And for this case, you can do it actually analytically, and these are this dispersion relation, so the relationship between the growth rate and the wave number. Sigma is essentially given by these formulas, which is essentially you have the point k equal to 0 is actually shifted. So essentially, if you plot now the growth rate sigma against the wave number, you see that as you change one parameter, the kind of instability that you get is that this parabola here, where the 0 point is missing, actually crosses 0, becomes unstable. And this actually happens, this point here happens all along this blue curve. And yeah, but since typically the cell and the systems that you are interested in are finite, this finiteness of the system essentially introduces a discretization on your dispersion relation. So you have, instead of being a continuum now, you have a discrete number. And you see clearly here that the first, and then you have the instability happens when this parabola, as you change parameters, this parabola essentially goes up. And you have the first points here crossing zero. But you see that the first points that actually cross zero are this and that guy, which essentially correspond to the system, to a pattern which is essentially fits just once into the system. So it corresponds to this pattern. Whereas the next one corresponds to that one, and so on. So what you see from here is that this instability is such that it tends to divide the system into pieces, because the pattern which is more the mode that is most unstable is this one, always, no matter of the size of the system. Yeah? And this is clearly this is from the Turing instability, where you get a well-defined wave number which doesn't depend on the size of your system. So in a way, this instability has the property that it can sense how big the system is and makes unstable just the pattern which fits once into your system. And with that, we can go back to the original system and look what happens there. And here again we have, so this area here is where we have the formation of domains. Here is this wedge-shaped area of bistability, the codimension 2 point here. And here I plot for 3 different cases the growth rates for this case, with the corresponding discretizations. And you see here as well that, for instance, outside in this region, the pattern is, the homogeneous pattern is actually stable, and it becomes unstable when you cross here. But what is, you still see a pattern here and a robust pattern here, because this instability is actually always subcritical. So even though the system is stable, you still see something which a pattern which arises subcritically. Okay, so this is all. I mean, this is what characterized the model that we wrote, and now we would like to, for the end, I would like to go back to experiments. So, as I mentioned you before, so the biologists are able to they tag to these two different genes with these fluorescent markers, and then they are able to measure the amount of the green and the red protein, and then they plot those protein profiles along the perimeter of the thing. And this is what we have been able to compare with the solutions of our model, where we put all the parameters that actually have been measured. And as you see, the comparison is rather good. And but this is not all. Also, the dynamics is also consistent. For instance, the times that it takes for this pattern to arise is rather consistent with the times that you see in the experiments. And for instance, this, actually, the experiments, as you see in this row here, so these are different snapshots. They are rather noisy, but it's still possible to see a nice comparison. And with this, I would like to go to a summary of the talk. So, what we studied was a mathematical model that describes symmetry breaking during cell polarization. And what we saw is that behind this symmetry breaking there is an instability, which is similar to Turing, but it's not a Turing instability. And this instability is such that it tends to divide the system into pieces. And also we saw that this instability is always subcritical, and this we can actually prove mathematically from the normal form that is always so. And this subcriticality somehow it gives you the property for the system to generate robustly domains. And that's it. Thank you.
about the symmetry break.
Any question or comment?
I was thinking about the symmetry break. You know, at the end is something that substances almost one side, almost to the other. There is no, you have not really considered a spherical shape, nothing to the forms. In that kind of situation, you see many other modes of phase separation.
Yes, but this is not phase separation. I mean, the point here is we didn't really want to write any model which
rich phase.
Sorry?
We have an A rich phase and a B rich phase.
Yes, but the logic is different here. We don't want to. I mean, of course, there are many ways that you as a theorist can think of putting up a system that reproduces these kind of things or does these kind of things. What we wanted to do was rather like a proof of concept. We knew a number of facts from the biology, and the biologists actually, when they came to us, they were interested in knowing: is this enough to get to observe what you see, or do we have to still keep looking for something else? And what I think what we can learn from here is that they already have it. They already have all the properties that lead to polarization. So it's like in a way a minimal realistic model which in one hand fulfills the seen observations and reproduces the overall experiment.
I think I have a similar problem to Raúl, probably because we have a similar background. But so essentially, forgetting about the biology, which is behind and which motivates all this work, what you are trying to describe is a separation in two phases. You call it polarization, symmetry breaking, and something is a mixture of red and green, and it becomes reddish in one side and greenish on the other side. And in your discussion, you proposed some type of one-dimensional model because you are looking at this on the surface. Now, this one-dimensional model, what we call the toy model, just to understand, as you describe it, is one in which is what one would write down for an Ising model with an external field because there is a symmetry breaking. Then, if you put that in one dimension and you have there an interface between the red and the green, this interface should move.
Yeah, but it doesn't move. What it means, the interface, that's a very good point.
So let me finish. So that's one thing I didn't understand about
the integral term is what essentially breaks the movement of the front.
Which term?
The integral term. So I had
now, Maxi, is with Kawasaki.
This guy.
No. This is okay. This term. Okay. Because this is okay.
This is the guy. I mean, if I put eta to 0, then you always get a front which travels and essentially it's.
Okay,
I mean,
so
one is the other, except in one particular case where the front is staying. I mean, that we all know. But also, look, in that case, you will always only expect to have fronts in the bistability region. But here you see that because of this guy, essentially, you have them in a much, they are much more robust.
Okay, so I understand this. I see now the importance of that term, and I didn't pay attention to that. But if I don't pay attention as I need to this term, then if I was going to describe some sort of separation in 1D in which I have a conservation law, then I would have a different type of equation with some which is not there. So again, I don't see very much what is the conservation law at the level of
it's a tricky thing because the conservation is not a.
Conservation is not there.
Because I already eliminate. I mean, I already use it to get to the. I mean, you have different. I mean, like you have one first conservation, which is the total amount of protein. All the proteins are conserved, but then you have each of the two proteins are also conserved, and that's what I use to get. Okay, I didn't get here from the conservation laws. I mean, this is just a phenomenological model distribution.
Because this is only the protein at the membrane.
This is the protein at the membrane.
Or the membrane, they are not conserved.
Yeah.
And the conservation comes through the global coupling in that global things. How? effectively, I guess. Okay.
Exactly.
We have at least two questions. Could you go to the next to last transparency? Yes, I don't see. If you compare the experiments and the theory, there is a clear difference. In the experiments, you have a green and red separated left and right, and in the other, you have a central block of green. So
Sorry, I was sloppy here. This is just. I mean, there is a. This is just. They are only looking to that.
Yeah,
the half.
So are you doing this with periodic boundary conditions?
Yes.
So where you show this? Because this is not the most unstable mode. This is the next one.
No. This is. I mean, this is only show here only half of the system.
No, but in you say that the instability breaks the system in two, and this is what is in the experiment, and this is not what is seen on the simulation. This is broken in three.
Why?
So something's happening with the boundary conditions.
Are
they periodic or what?
They are periodic, of course.
It's a ring.
Okay.
It's a ring.
Because
it's a membrane. You assume
then
we are periodic.
In a periodic ring, then you cannot break the system in two. You can. The first instability is breaking the system in three. Well, okay, you can break it. I look at the difference between the experiment and the theory. Now it's clear because in the experiment the boundary conditions are not
so
periodic.
you should look at this.
They're showing on the class.
This is how. I mean.
No, in the experiment there are no periodic boundary conditions.
Yes, actually it's a surface, which is more, that's my second question.
But then they are plotting.
You mean that the LED, no, you mean, okay, you mean that you're, so it's true that it's a periodic,
yes.
you're considering that in a one-dimensional object which is periodic.
Yeah.
Okay. So I didn't notice that. I was thinking on the,
you
could something like something on the horizontal direction on the above pitch, but it's not the horizontal, but the circular direction.
You could also take half of the perimeter and take zero flux. That also works. But I mean,
okay. This is what I was thinking of.
Okay.
Okay.
No, I was not clear.
Okay. The next question: in the model, you have a parameter alpha of the non-linearity. You have nothing.
Yeah.
You have any incremental constraint on that, or whatever you need? Or?
Yeah. In a way, yeah. No, you need they can be equal. Actually, we take them equal for.
You put the two parameters, alpha and beta.
Yeah.
In the equations, yes.
Well, we have done many different tries with that. What did I? Anyway, I had that have an extra slide. Oops. So here I put different alpha and betas, and essentially what you see is that you have a constraint, which is you need that alpha or beta are one of the two should be bigger than one. But beyond that, it can be anything. So because you see that essentially the bistability area, so this last little thing here is when you put in alpha and beta equal to 1.1, so it shrinks and shrinks, and essentially for one it disappears altogether. And yeah, beyond that, we took in the paper thing 2 and 1 because of some biological reasons about how the biologists think that this interaction goes on. Yes. But yeah.
On your model is in 1D, but in fact the cell must be a 2D surface. So actually, to be
essentially, well, even 3D. I mean, it has a
the surface is 2D. I mean, the topology of this, so this defect, this structure is created is you see a bump in one system. I guess in the cell is a line.
Yeah.
Through the whole.
Yeah. It's a cask. I mean, it's of course. I mean, you have. It's a surface, as you said. So it's you have like a spheroid.
Yeah, but then you qualitatively it doesn't change much. Or it does, I don't know.
No, it doesn't change much. We have done the simulations.
On the 2D surface.
The 2D surface. It's just messy. And
you have curvature effects.
Yeah, you have some. I mean, it's not exactly the same, but it's actually very similar. But rather more problematic is what you assume from the bias. because okay, this is a very tricky issue, which is we are assuming very fast diffusion, lateral diffusion, but also, I mean, in the bulk you have a 3D diffusion, and this is actually a quite tricky point. And for the membrane, we saw that, I mean, between the 1D or if you do that in a spheroid, it doesn't make much of a difference. But with this other thing, is not problematic.
More questions? Comments? Now let's thank our speaker. A coffee.
Yeah, we'll have some coffee, but in the meanwhile, we can have to look at the DVD.
I forgot it. Ernesto wants to